Good morning, everyone. Thanks a lot for coming to this public lecture of our colleague Jakub Hofman. I'm very happy that we are here in such a large number, and I would like to welcome you and also welcome our colleagues that are online. we stream this lecture for the members of the professorship committee that has been nominated by the Faculty of Science Scientific Board. And the members are myself, Professor Holoubek, who is here, and then three colleagues outside of university, Professor Shantruchkova from South Bohemia University, Professor Froos from Charles University, and then Professor Kies van Gestel from Vrije Universiteit in Amsterdam. This lecture is a part of professorship procedure. Some of you are not Czechs here, so I will very briefly explain. This is a kind of demanding procedure when the applicant have to prepare dossier for assessment, and one part of the process is this public lecture, where he or she should demonstrate both the expertise in the field and also the capacities to be able to translate, teach, lecture. So this is the goal for today. And I would like to very briefly introduce Jakub now, and then he will have all the floor. He received his PhD in 2003 from environmental chemistry, and then in 2011 he habilitated, meaning that he became a docent in the Czech system with the topic on contaminants in soil and their effects on biota. He has a lot of experiences with teaching for many years here and also at Charles University. He teaches ecotoxicology classes, soil ecotoxicology, microbial ecotoxicology, both in Czech and in English. He has supervised 20 bachelor, 23 master, and 8 successfully defended PhD theses. Now, as if I counted correctly, there is 6 or 7 students under your supervision now. And he published over 52 papers in Web of Science that received, and I checked this morning, more than 500 or close to 600 citations, which all qualify the expertise in soil ecotoxicology very high. So I think that we will get entertained today by a quite hot topic, and that's all. Jakub, please, the floor is yours.
Good morning, ladies and gentlemen. Good morning, colleagues. Today I would like to talk to you about a topic which I selected from our research topics because I think that it's very novel, it's very interesting, and it's emerging, and it's nanopesticides. Before I will start, I would like to thank, maybe not very usual, but I will do that at the beginning, to all my colleagues from our research group, because without them, this research and also other research topics of our group would not be possible. The junior researchers, PhD students who did a lot of lab work, a lot of experimental work, contributed to the writing, and without them, these projects would not be running. In my presentation, I would like to start with some short intro about soil and its degradation. Then I would like to follow with introducing our research topics. The main part will be about nanopesticides, as was promised in the title. Then I would like to show the first pilot study of our group, which was about the nanopesticides, and then I will finish. At the beginning, I would like to go back to about 15 years ago when we started to do research on soil. Why? Because soil is very important. It's delivering the crucial ecosystem services for environment and for human society. It's important also for the sustainable development goals, its protection to manage the soil sustainably, and also it's contributing to one of the planetary boundaries. Unfortunately, the soils are facing today to a lot of stress. They are degraded. Considering the total soil area of the world, about 8.7 billion of hectares, we have about 23% of this land degraded. And what is worse, that when we consider just arable soils, it's about 38% of arable soils in the world are degraded from various reasons. So it means that we are losing the soil, and some types of the degradation, like for example sealing and erosions, are not reversible, and it means that every 3 seconds we are losing the area of potentially fertile soil, which is equal to a football pitch. Combined together with the increase of the population in the world, there will be like 9 or 10 billion people on the earth in 2050. Just when compare this, the area of the arable land per one person in the earth is decreasing, and this is not possible to, it will be not possible to feed all people very soon, because we are not able to produce food any other way, or mostly any other way, just using the fertile soils. One of the degrading factors of the soils is contamination. By the size, it's about just 10% of the degradation among the erosion and sealing and so on is contamination, but when we express this as a number of sites, it's pretty high. Just for Europe, it's estimated that there are 2.5 up to 3.5 million contaminated sites. About 10% of these are so contaminated that they require immediate remediation. This is also generating very high cost for the society, not connected just with the remediation, which is about 2 billion euro per year, but also with the decrease of the cost of the land, health, increased health care, and so on. In Czech Republic, just for information, we have up to 10,000 contaminated sites. So this all contamination from various compounds, from various processes of the mankind, of course, severe impacts not only on soil biota and biodiversity, but also directly on human health. And what is very important, that these two are somehow connected through ecosystem services, which I mentioned at the beginning. Because we feel that this is very, very crucial, state of art we are now with regard to soil, we think that also our research should contribute to these issues. So our main goal of the research of our research group, which might be called Soil Environmental Chemistry and Ecotoxicology, here at RECETOX, the main goal is to investigate complex interactions between contaminants, soil and biota. We do studies of fate and bioavailability. In the past we focused on the persistent organic pollutants. Nowadays we more focused on currently used pesticides. We do soil ecotoxicological testing, not only routine testing of the pure compounds, but we more focused on real materials like inputs to soil, like sediments or sludges, and assessing various anthropogenic impacts on soil quality. We also have performed a lot of monitoring studies, assessing again some activities of the society. We monitored around industry, we have monitored around highways and so on. For that we also establish a battery of the methods which we can call microbial bioindication of soil quality in the past. And last but not least, we also focus in some project, and the first supported project we have got just recently. We focus also on some new technologies, mostly connected with the waste processing and treatment of the soil and climate change mitigation and so on. So altogether, we want to focus not only to deliver the new research results, but we also very like to somehow contribute to the soil protection to the society, which I think should be the mandatory thing for every scientist. So if I just briefly mention the projects in last 10 years which were contributing to these research issues, it was the bioaccumulation and studies of the bioavailability of persistent organic pollutants. Then we also had several, let's say, applied science, applied research projects focused mostly on contaminated soils, either forest soils or alluvial soils. As I said, we have also monitored the environment around industry, particularly around cement factories. And when I would like to also mention the current projects, it would be the fate and availability of currently used pesticides, which is the project which recently finished, and we are finishing all publications from that. We have also two projects which are somehow connected with the biochar material used for improvement of soil and its interaction with the pesticides and chemicals in the soil. Then we recently got funding for the biotechnology for conversion of waste of spent grains by Hermetia illucens larvae. This project will be presented in few weeks. And the last project, which is also mostly connected to today's presentation, is the nanopesticides in the environment, understanding their fate, bioavailability, effects, risk in soil and water. So now I would like to talk about nanopesticides. Just put it in some broader context, I would like to remind that we have pesticides as a tool for increasing the crop yields for the humankind. And as you have seen, on the slide, which was about the increasing world population. We need that. We need our fields to deliver us more yield. But on the other hand, the use of these pesticides is increasing. It's approaching to about 4 million tons per year in the world, and Europe is contributing with about 500,000 tons per year, and the most pesticides are used every year in China. It's almost half of the all pesticides over the globe consumed per year. This market is very huge. In 2025, it will reach about $9 billion, and unfortunately, there is also happening something which is not very nice from the viewpoint of the competition between companies. These companies are merging. So today there are just 4 giants or 3 giants which are leading this whole market, and of course the influence on this development is lower. On the other hand, I would not like to sound that I'm cursing these compounds, because they are really helping us to save the crops. Not only their amounts, without pesticides, something about 20 to 40% of the crops will be lost, but they're also keeping the quality of the food. Not everyone knows that if we omit the residues of pesticides from the food, there will probably appear the residues of mycotoxins, for example. But there is, of course, a lot what can be improved. There are some assessments that using such huge amounts of pesticides could be reduced with keeping the same efficiency. Nowadays, only 1 to 10% of the pesticides are reaching, applied pesticides are reaching their targets. The rest is just useful amount, which then form potential threats to ecosystems and human. So it means that the pesticides consumption must be reduced, but keeping still the help to keep the crop for the increasing number of people. So it's forming a big space for new products, novel technologies to improve efficiency and reduce impact on environment and human health. And this is the real goal for the nanopesticides, why they started to be developed. Firstly, I would like to define somehow what are nanopesticides. So it's probably quite logic that these are the pesticides which are small, and they contain the particles which are below 1 micrometer, but sometimes in the legislation of nanomaterials there is another threshold listed, and it's 100 nanometers. So this is still kind of discussion about this threshold. And there is a literature, yeah, by the way, I forgot to mention that the references in the presentation are listed on the web page for those who are interested, and the link was on the outline slide. So there is a literature which is stating that it's not just the particle size what is defining the nanopesticides, but there should be also some other things fulfilled, like for example that they are intentionally present, these particles, that they are delivering some new novel properties when compared to the conventional conventional size, and that also if the product is just named somehow nano product, it should be considered and examined for these nano related properties. Generally, the goal of these nano formulations are very positive. It's in general decreasing the environmental impact of pesticides, and especially amount, because when we see this curve, this is current type of application we have. It's large amount of pesticides applied, quickly decreasing, and only small amount of the pesticides is reaching the targets. So this should be changed with the nanopesticides, especially by the property which is called slow release. So we will deliver a small amount to the ecosystem and somehow guarantee the continuous exposure of the pests. Another goal is specific and targeted application, that some carriers are developed to deliver very specifically the active ingredient only to some environment, for example with some particular pH, or to some specific organisms, which is expected, of course, will decrease the hazard for the people who are working with the pesticides, and also the risks for the non-target species. So in general, we may say that we are going from the conventional pesticides, which are high volume, low value applications, to low volume and high value applications. From the technical point of view, there are a lot of benefits of these nanoformulations. So firstly, the problem of most pesticides currently is that they are not soluble, which is complicating their applications. And sometimes also the drawback of such compounds is that they are quickly degradable, for example, by light and so on. So a lot of technical improvements when these compounds are nanoformulated. And firstly, it's bringing better technological properties like stability in tank, better adhesion on leaves, and so on. And no need, in conventional formulations, there is high volume of solvents, surfactants, oils, which all are contributing to the environmental pollution. In a new formulation, this is not necessary. As I mentioned already, higher stability and resistant, which should protect the pesticides after application against their leaching, runoff, and so on. Higher efficacy, this is something really needed, and it was already proved, as I will show later, for some new formulations that they are really more effective to the pests. And as I mentioned already, controlled slow release, which should guarantee some constant level in the treated environment. Sometimes also are developed very complicated nanoformulations comprising more components, which should guarantee some very accurate and specific application, like key and keyhole, for specific targets, time conditions. There are also formulations which destruct themselves after being active in the ecosystem. Higher degradability, this is the case, that there is kind of catalyzer involved in this nanoformulation, and the pesticides stop functioning after some defined time period. And these nanoformulations are also enabling the use of new active ingredients, for example, like plant derivatives, like some specific oils, which are again very hardly to deliver to the pests when they are not on some carrier. So these carriers can deliver these plant extracts, plant oils to the pests and replace completely the chemical pesticides with their natural analogs. There is a lot of types of nanopesticides. I'm not going to talk about all these. There are always the type named in this box, and there are just examples from the literature which compounds have been already tried to formulate in this specific type of the carrier or type of a nanopesticide. Probably the most frequent and most mentioned in the literature and also in our focus are these nanocapsules and nanospheres, which are really the carriers, particles from organic polymers of both synthetic and natural origin, bearing the active ingredient. And these type of compounds are really delivering very new and novel properties to the pesticides. Some others which can be mentioned are the carriers which are from inorganic, for example from silica or clays. And in general, we may conclude that this, the type of this formulation will definitely influence the environmental properties and toxicity and ecotoxicity of the pesticide, which is bound on these carriers. And also, according to already published approaches of the risk assessment and evaluation for these nanopesticides, it is also crucial key to these concepts. So at the beginning, we have to distinguish what type of the nanopesticide it is, and based on that, also the possible risk and possible behavior in environment, and the assessment approaches are then differentiated. There is, without any discussion, boom of nanopesticides. Sometimes it's tricky to find the right patents, right literature, because it's not always called nanopesticides. You may face nanoformulated pesticides and some other words. So it's a bit tricky to find the right literature about that. But without any discussion, it's increasing, probably as all literature is increasing, but also in a relative way. The papers about nanopesticides, there are also the first books just about nanopesticides. There are already some products on the market which contain the nanoparticles, and there was also a review of the patent databases in 2013, and it stated that there are more than 3,000 patents which are somehow describing nanoformulated pesticides. So it's a really huge increase. And if this application of nanopesticides will start, it means that they will be really used, these patents, in real products and will be introduced to the environment, together with the amount of pesticides we saw on the previous slides. It will be the first intentional diffuse input of engineered nanoparticles to the environment. Because the other nanomaterials in the environment are not intentionally there. It's like pollution from the industry. It's connected with the huge boom of all nano in agriculture, not only nanopesticides, but also nanofertilizers you may find, nanosensors and so on. These are the keywords which are really frequently appearing also in the project world. You may find a lot of European projects from the recent years which are focusing in so-called converging technologies, which are the combinations of technologies, informatics, robotics, and things like that in the agriculture. Even now, there are also some companies and project teams which are developing the drones who are applying very specifically the pesticides, and all that together is called precision farming. We as a group were invited in 2 projects in last years which were focused on precision farming as a combination of robotics, drones uses, remote sensing, assessment of the fields, application of the nanopesticides, all together in one project. Of course, this is generating a lot of critics. Some of them is just fear, but some of them are reasonable, as I will show you in the next slides. Why to pay attention to the nanopesticides? The first thing I would like to say is because they are nano, so it's nanomaterials, as all engineered nanomaterials, engineered nanoparticles, and the same awareness should be also in the case of nanopesticides, maybe multiplied by the fact that they are really intentionally applied and they are really close to the food and feed. this technological development is much faster than getting knowledge about these nanomaterials. And there was also a very interesting study comparing the money which are each year delivered to the research for the nanomaterials, but to the research of, let's say, technical development of these nanomaterials, compared to the money in USA, to the money which were for the research and development of the impact of these nanomaterials. And you see clearly this inequality in that. So we don't know a lot about nanomaterials still, despite there are in a lot of hundreds of products of our daily use. I just go to the supermarket and take picture of that nanoliquid for a car glass. And there are really in many products also connected with the food and hygiene. The key question probably in this area is if the nano, it means that the chemical is somehow formulated in particles which have nano size, is changing somehow the behavior, the toxicity, the risk of these chemicals. And this is still ongoing discussion at the level of the authorities, EPA, ECHA in Europe, EFSA. They are discussing this. They are somehow trying to do the data mining from the literature if this is right. And most of the studies answer yes. That means that nano character is changing significantly all these processes, and so this nano particle attribute should be somehow involved also in assessment procedures, evaluation and authorization of the chemicals, including pesticides. And just two examples from the literature, that smaller particles, of course, everyone knows, are going deeper when we are breathing the particulated matter, and also that they are somehow easily transported through biological membranes and barriers. So it's also expected that for nanopesticides it should be affected, these processes will be affected: environmental fate, exposure, bioavailability, ecotoxicity, toxicity, health effects. This is a scheme for Australian guideline which was published in 2015, and it's showing in the red box all processes of the risk assessment scheme which will be probably affected by the nano character of pesticides and nanofertilizers. Still, there is plenty of unknown. What we want from the viewpoint of the regulation is somehow judge between two things: the benefits which these new technologies, these new formulations can have facing the problem of pesticides, which I illustrated, and the benefits the disadvantages, the drawbacks, which are the new risk which we haven't known from the conventional pesticides. New risk which are related to the nanoparticles. As we did extensive literature review, it's apparent that these papers which have been published, so that scientific knowledge nowadays mostly is focused on the pure chemical development of these new formulations, characterizing the particle size, if that was successful chemical production process, and their new physical chemical characterization. This is dominant number of papers. Only very small fraction of these studies are somehow assessing if the goals I listed, the goals of the nanoformulation, which should be beneficial for the environment, are really somehow fulfilled with these formulations. Only very small fraction has some toxicity testing, and now I'm speaking about the toxicity towards the targets, towards the pests. Only small fraction, and much lower fraction. In fact, only these listed papers are somehow focused on the studying of E-fate or their effects on non-target organisms. And that ratio here, you can see on that slide with the development of the VOS publications. So it means that we have several hundreds, maybe approaching 1,000 publications about nanoformulated pesticides, but only few are about their effects on organisms, their fate in the environment. Despite significant impacts of nanoformulation on fate and ecotoxicity were observed, the mechanisms involved were little investigated and remain poorly understood. So even those few papers, they just measured and stated there is a difference, but nobody knows why. And this is also one of the reasons why we started to do this research, because we want to know the mechanisms behind why nanoformulated pesticides has different effect, different effects than conventional pesticides. Because there is still plenty of unknown, it's very hard to then assess the risks, somehow regulate the use of nanopesticides and trade off the risk and benefits. And because they are still very fastly developing, we feel that it's also urgent need to increase such scientific knowledge. And it's priority not only from the viewpoint of the soil and water quality, but also from the human health. The new results of such research can contribute to all involved in this process, to the formulation scientists, because they need some feedback that the formulations they are developing really bring these new important and beneficial properties for the pesticides. And also to the risk assessors and regulators, because then they will know how to assess what properties should be measured newly when compared to the conventional guidelines and so on. Just to quickly summarize what is changing if the pesticide is formulated in nanoparticles. So firstly, there will be the physical chemical properties of the nanocarrier, which are coming into the play and play key role. You can see here some list. So of course the particle size, particle structure, surface properties and so on, which are now important for the environmental fate. Secondly, decreasing the size, we are coming to colloidal particles. And colloidal particles have very special features, very apart from the conventional Newton physics we know. So they are starting to behave very differently. For example, in the liquids, they are not sedimentating and so on. So many environmental processes will be affected just by the different size. New processes will start in the environment. And third point, probably one of the most important, is that apart to the kinetics we are studying, we know the kinetics, for example, the degradation kinetics, the biological uptake kinetics, but here there is a third kinetic added to all these environmental processes, and it's a kinetic of release of the active ingredient from the carrier. And this is something for which we also do not have a lot of working tools nowadays, and this process will probably influence also the decisions when the authority want to assess these environmental processes and risks. And from the viewpoint of effects, just very simple logic. If the nanoformulation is developed to be more toxic for the targets, then it's a logical question and a logical conclusion that probably many features which are going to this goal will probably increase also the environmental risk and toxicity to the non-targets. just considering slow release, which I show as something positive, but now we can view it from the other way, that the exposure of the non-targets will be much longer and higher in that case. And there are studies which are showing the differences of ecotoxicity. Very frequently, they are really confirming that also for non-targets, the nanoformulated compounds are more toxic. But as I said, we don't know why. If there is, for example, higher uptake of these compounds, but the specific toxicity remains the same, or if it's also somehow influencing the toxicodynamics, we don't know. And there are also toxicity studies. These results on mammals or mammalian cells are not so clear. There are some studies who are saying nanoformulation is decreasing toxicity, and the others the other results. Very nice publication was recently published by Melanika, showing, taking all the studies which have been published so far and somehow summarizing the prevailing trends. And it's apparent that this ratio here shows how the nanoformulated pesticides increase when compared to conventional formulation and compared to pure active ingredient. And we see that the line here is 1, and so it's clearly apparent that most of the nanoformulations increased the toxicity towards pests, towards the targets, but also most of the formulations increased, for example, the persistence in the environment compared to active ingredients. So all this knowledge together, it's apparent that partly we need to add more knowledge about this phenomenon, but partly we can already draw some conclusions. So these nanopesticides are also the challenge for the regulation and assessment. Probably the currently used approaches defined by EFSA for pesticides, defined by ECHA for chemicals, probably will be sufficient in the case that the nanoformulation is not affecting the environmental fade-in effects. That's logical. But knowing the types of the nanoformulation, we already know that this most probably will be the case for example, for nanoemulsions. Fortunately, the most of the current plant protection products on the market which contain nanoparticles are the nanoemulsions. So we may think we are safe for now. But these procedures, these regulations will probably be not sufficient for the nanopesticides which have the type that there is kind of carrier, an active ingredient which is bound to the carrier. For such kind of nanopesticides, these ways how the pesticides are assessed and evaluated will not be sufficient. So there is, of course, a need to update all these methodologies, these guidelines, these processes, and these organizations like EFSA, EPA, they know it, but unfortunately they don't know how to do it. There are a lot of methodological challenges which they cannot solve, of course. They need more scientific and research outputs for that. Not only for nanopesticides, but our knowledge about nanoparticles in general is limited. Also, we are going to be handless sometimes because the methods we have known so far, like OECD test, OECD conventional methods for KOV determination, for soil sorption determination, and so on. All these OECD methods can hamper, can be not useful when nanoparticles are in the system. The results which they will generate can be completely wrong because, for example, of aggregation or disintegration during the method. There is very big challenge for the toxicity testing we are able, and everyone knows that during the testing the toxicity, you have to check the concentration of the tested chemical. That is what we know and what we do. But for the nanoparticles, nanopesticide, it means that you don't need to check only the chemical concentration, but also you need to check if the chemicals or the particles are still there, if they are aggregating, non-aggregating, what is the ratio between the released and bound chemical. So it's much more complicated. Some established theories will completely lose their validity. There are new processes coming to play, like quantum effects, particle physics, which is replacing or added to the solute chemistry and so on. So new methodologies, new instruments, new approaches, new interpretations. Yeah, like in case of solubility, because you can keep a lot of pesticide which is completely insoluble in water, bound on particles, very homogeneously in the water solution. So is that solubility or isn't? We lose the interpretation of what we know. It's apparent that experimental protocols must be adapted for all kind of steps in the evaluation, for the extractions, analyses, toxicity bioassays, environmental fate methods, especially to distinguish between solute and particle chemistry, to distinguish between bound and free pesticides, and between loaded and free nanocarriers. So this is probably the three most important things which must be considered in these new methods. So release kinetics is probably the keyword which is now really in play in a lot of these studies. Based on this conceptual view, there are also first birds of the assessment methodologies. In Australia, there was developed the rational scheme for the nanopesticide assessment, which is based on the judging what kind of pesticide and nanocarrier is there. If this association is very strong, and this nanocarrier pesticide interaction complex is long-term, it has also very important consequences for the evaluation. In that case, probably also toxicodynamics will be affected if the complex of nanocarrier and pesticide will be short-living. It's affecting maybe toxicokinetics, and if it's very fastly disrupted and the active ingredient is very fastly released, it can mean that it's not affecting at all toxicokinetics and toxicodynamics. So after application to the environment, the pesticide is behaving the same way as the conventional form. So that's just the example of this decision table. So in Austria, they handled this issue pretty well. On the other hand, in Europe and in USA, the authorities responsible for the pesticide regulation, they are not very far with that. They don't know, and we just knowing what I presented so far, we can draw some conclusions or recommendations how the guidelines for these pesticides should be changed. So there is a need to identify these particles, or identify the products which contain these particles. We have to involve some analytical tools into the registration process to determine which plant products contain these nanoparticles. And also, if already approved active ingredient has a new formulation which is nano character, it should be again evaluated as a new registration, which is not the case for the conventional formulations in current system. Specific properties of the nanoparticles should be involved in the assessment, and also these properties must be placed or must be reported in the dossier, which is not mandatory now. Some uncertainties should be involved when pesticide contains some nanoparticles. And now the formulations are tested for the ecotoxicity, but E-fate is assessed only for the active ingredients. But in case of nanoformulation, we have seen that a lot of processes also from E-fate will be affected by this. So also the testing whole formulation must be involved in E-fate assessment. There are some first approaches. The European way how the plant protection products are assessed are so far not very adapted for the nano-formulated pesticides, mostly because of this, that when the pesticides are evaluated in the Europe, the E-fate is considered only based on the active ingredients and not on the formulation. It's supposed that the formulation of the pesticide, whatever it is not affecting the environmental fate. That it's affecting the ecotoxicity and toxicity, but not E-fate. So this is something which must be changed for the nanopesticides definitely. And something like that is already placed in the biocide legislation, so there must be can be also some inspiration from that. And very new, very recent EFSA guideline, which is firstly very general. It's not just about nanopesticides, but it's about all nano science and nanotechnologies in food and feed industry, food and feed chain. But there is Appendix E, which is solely about some conclusions for nanopesticides. And there are some first things which I have already listed as recommendations, and it means that that if the pesticide is containing some nanocarriers, there should be a list of additional properties. There should be specific nano-specific aspects which should be considered in the testing, and also that the nano aspects should be considered in exposure assessment. And this is really something new, that formulation will be somehow considered in the assessment of the E-fate and exposure. So summary, which has been published in our review paper of the all plus and minuses, which can be summarized from the available literature of the nanopesticides. And the conclusion, in fact, is kind of balance. Yeah, we have pluses and minuses of these formulations, and without any discussion that we still are lacking the research outputs to decide, and also decide type, nanopesticide type specifically, and probably also compound specifically. So we need a lot of research. And this is the bridge, how I'm coming to the end presentation of our Gotcha project, where we want to research these nanopesticides and add the knowledge about their environmental fate and ecotoxicity. And that's the theme of the project. Again, I would like to thank these guys here. And the goals are pretty simple, and surprisingly, the first goal is not to research the nanopesticides in water or in ecotoxicity, but methodological, to adapt, to develop and verify the methods who can be then used for the nanopesticides, and especially to distinguish these things which I already mentioned, the solute particle chemistry and so on. Because we found that what we expected will be working is not working. Determination of the sorption, for example, and all that stuff we know is influenced by this nano character. The second, after this starting phase, we want to elucidate more not just conclude that there are differences, but elucidate the mechanisms which are responsible for the differences of fate, behavior, and bioavailability of nanoformulated pesticides. and also to investigate the changes of toxicity and risks of these nanoformulations. And as an example, as a pilot study, I would like just to show very, very quickly the results of the recently finished study, which was comparative study. This one was not going into the mechanisms yet. That will be the task in new project, but this is just showing that we have very good proof of concept that there are differences between nanoformulations and conventional formulations and pure active ingredients, even at the level of the ecosystem or microcosm level. So what were the goals? We just wanted to study the effect of pesticide formulation on fate, behavior, and bioavailability, comparing the pure pesticide, nanoformulation, and conventional formulation. We also involved 2 compounds just to compare if this is a general conclusion or it's compound specific. And we also involved just 2 soils to see if there might be some difference in soils of different properties. What we studied: chlorpyrifos as an active ingredient, tebuconazole as an active ingredient, then conventional formulation of chlorpyrifos, conventional formulation of tebuconazole, and nanopesticides. We used first carrier. We used both nanoformulations were from our Brazil colleagues. Soil lipid nanocarriers and polycaprolactane carrier, and both carriers were used for both compounds, so 4 nanoformulations altogether. The concentration, how they were able to prepare these nanoformulations was pretty high. You can just check with the solubility that these formulations are really changing the properties of these compounds, because they are in fact in water. And a very efficient preparation that these compounds were from 95–99% really associated with these nanocarriers. We added all these compounds to the soils, all to have the final same concentration of the active ingredients. Then earthworms, plants were seeded, particularly Lactuca sativa, and after some time points, 1 day, 3 weeks, 6 weeks, and 12 weeks, we dismantled these microcosms and separately analyzed leaves and roots, earthworms, soil, and we also employed a passive sample SPME fibers with PDMS as a sorbent. After all analyses, we got the results. These figures are showing the real outputs of the measurements as concentrations in soil, and the modeled curves according to first order degradation kinetics. And from these results, we see that nanoformulation, at least in some cases, really significantly change the degradation in the soil. Especially nanoformulation 1 on solid lipid nanoparticles slowed the degradation in some soils. Then we saw also the differences between the LUFA 1 and LUFA 2.4. This soil has a higher organic matter and higher clays, so it's apparent that it's promoting microbial communities which are degrading these pesticides faster. And there were also differences between the compounds, the chlorpyrifos being more degraded than tebuconazole. Here we used toxicokinetic modeling. We modeled the concentrations measured in earthworms with toxicokinetics, which is also considering the degrees in soil. So that's the reason why we have this kind of curve with a peak. And overall, it's not necessary to go to detail, but overall, we again found that, for example, nanoformulation 1 is significantly changing the uptake to the air forms. In fact, it's slowing slowing the elimination. So finally, the worms have higher concentration of these nanoformulations in these pesticides in their bodies. Again, we were able to compare also soil, that the bioaccumulation from the soil 2.4, which has more organic carbon and clay, was lower than from the other soil. Similar conclusions can be drawn for leaves. There was even no uptake of chlorpyrifos at all in this soil, because probably having too high organic carbon to allow these pesticides go to the pore water and to be uptaken by lettuce leaves. And again, some differences between formulations and pure active ingredients, which is the solid black line. And we also tried to employ these passive samplers, as I said, these SPME fibers, but unfortunately it only showed that there was difference between soils and between compounds, but there was very inconsistent observation between the formulations, so we cannot conclude anything for the pore water concentrations. How these nanopesticides are changing this parameter because of very high variability and also some cross-contamination we found. To conclude this study, I would like to say that despite that this experimental design was pretty robust, it was a complex environment, and also in some parameters we observed pretty large variance, so something can be of course improved for the future experiments. But despite all these things, we really observed significant indication that nanoformulation changes the soil, the fate in soil, it means the degradation and bioavailability to earthworms and plants. Of course, this is kind of proof of concept, as I said, because it's opening the space for the very, very large research of the detailed processes and mechanisms why we observed these differences. Especially, we will need some methodological improvements, more time points to improve the kinetics modeling with lower uncertainty, better working passive sampling technique for the pore water, and also some parallel measurement of the release kinetics of active ingredients. This will lead us to the mechanistical understanding of what is going on. Then we would like to extend these studies, or in fact we want to start with the aquatic environment, which is much simpler than soil, and then we will go back to these soil level studies. And we would like to link in the project the bioavailability processes, bioaccumulation, and ecotoxicity also. And we will also employ another models of nanopesticides, employing another active ingredients, another type of carriers. So that's a lot of to do. At the end, I would like to thank to all projects which were used to perform this research. I would like again to thank all colleagues from the group which significantly contributed to all that research. Then I would like also to thank to my beloved and thank you for your attention.
Thank you very much for a very nicely structured and illustrative presentation. Yeah, now I would like to ask you for your questions. I will, because there are colleagues hanging online, always repeat the question so they understand what it is about, and Jakub will address it. So, any questions?
I have one. There was something simple I didn't understand. When we are applying pesticides, like except when it comes like really when they are encapsulated into the seeds, but I'm speaking when we are about aerial spraying. Then you said something that the nano formulation will help to reach like release the target compounds, but it's known that smaller the size of particles, longer the particle will travel in the air and will be less keen to deposition. So I'm really surprised about this result. I would say for me, when it's applied in the air, smaller the size of particle is, more likely or higher will be atmospheric resonance time.
Okay, so I will very briefly repeat. Basically, the question is about the aerial application of the nanopesticides and their deposition properties related to the size of these.
I have no answer to that. I don't know. It's just supposed based on the general knowledge about the particulate matter in the air. Small particles are, of course, transported to longer distances, but that was the picture I showed. It's just expectation, because nobody, in fact, has studied this. But why the nanoparticle bearing the active ingredient should behave differently than any other nanoparticle? So if you know if it's silica, that's the same. We have a lot of silica particles in the air, probably, in this windy weather. If it's some organic nanocarrier, I don't know if we can find in particles in the air something similar, but that could be probably extrapolated, these conclusions. But there are no studies, particularly about nanopesticides in the air.
Thank you. Other questions?
Yeah, thank you very much for a very nice presentation. Yeah, it raises a lot of questions actually. Like, of
That's
course,
good.
that needs
We
to
have
be answered.
something to investigate.
Yeah, one thing was really about like the general thing. Is it then really? Do we really know it's beneficial to use nano pesticides in general, or is it linked a lot by the trend? To use nanotechnologies in general, because like I really agree with this question. I was wondering how that could actually affect the distribution on the soil. That's one question. Then another question is, since you have shown or discussed that actually the nanoformulations have been shown in some studies that they are like more toxic for the target organism, but as well for the non-targets. But then for the risk, like environmental risk, if they are more toxic to the target organism, then you can use less. So the less dose, application dose. So then the amount applied, is it going to be the same, or is it actually the situation for so the situation does it really make difference then for for the environment and towards like decreasing the risk for non-targets, because that's one of the benefits, right?
Yes.
That should occur.
So before Jakub answers, I will again repeat it for our colleagues. So the first question was basically about the benefits of nanopesticides and their relationship to the general trend in the society, if I understood, because now we are in the nano period. So is it really like a benefit, or are we heading there because it's a kind of trend?
I will answer with the history of these nanopesticides from the viewpoint of the perception by the society. Like 10 years ago, the companies were labeling the new formulations as a nano something, and it was stated in the leaflets for this new formulation that it's containing nanoparticles, so it's better, it has new properties and so on. Like 5 years ago, it was completely different, completely reversed. Nobody wants to mention in the new formulation that there are some nano things inside. So the perception of the society changed, and now there is more or less, I would say, the panic from nanoparticles. So the new products, you don't know if they are containing nano, because they are not obliged to manifest that these new products are containing nanoparticles. and that's the need for these authorities to check it, because they will not mention it. They just may state that there are new properties of that formulation. And also I would like to mention that the real problem of the pesticide world is that the number of approved active ingredients is very decreasing. So the whole pesticides development science in fact is changing to the formulation science. So what they are doing is to using this limited number of the active ingredients, but formulate them somehow differently to increase their efficiency and stability and properties and so on. And this will be worse and worse. There are lists of the compounds which are waiting for the disapproval. And the second question? The
second question, if I understood correctly, was about usage of nanoparticles, because as you described, there is a kind of benefits if you can use a lower amount of this nanoparticle and lower amount of active ingredients. Then it also affects the exposure going down. At the same time, they might be more toxic to pests, also to non-target organisms. How this balances?
That's exactly what you have shown, is the way how all these pieces of the puzzle should be put together. Like if you can use less because it's more toxic to the targets, of course that just from the viewpoint of the exposure, it's better to the non-targets. But if at the same time the specific toxicity is increased, I don't know why, maybe they are uptaking more because it's more mobile, bound on the particle, then it's compensated and nothing changed. So you have to really find a way how all these processes put together in the final risk assessment, and there might be the decision if but I think there will be no time point where somebody can say all nanopesticides are in general better than conventional pesticides. It will be very specific by the type, by the compounds. So, but these informations must be here as a feedback to these developers, because they have some dreams that they will develop something which is safer for the environment, and they need the correct studies to know if they fulfill their expectations.
Thank you, Kuba. I learned, unlike for other technologies in the past, which have been also introduced without a real assessment, that here we need also expensive technologies to do surface characterization and solid particle characterization, which, unlike in the past, is not established with the manufacturers. They don't have these technologies, and they have no need to really apply them. They can just sell a new product, trial and error, without really characterizing their product. So, and also the government has not these technologies at hand. It is a very costly characterization of a new technology which nobody does. The manufacturer does not do it. The government has no, only the science has in principle the potential to do it, but needs a lot of money to do it. So I see here a dilemma which was not there previously. When new technologies came up, the technology came with the manufacturer and could be forced by legislation to please apply it. But now, the environmental concern side is forced to prove with very costly technologies that there is a risk.
Yeah. So if I can repeat it a little bit again, in fact it was a question or maybe comment on the use or lessons from the past when the technologies have been used in the past and they needed to be assessed before they have been used. Now we are in the little different situation because the technology is out, and now if I understood correctly, basically the producers kind of are pushing the society to prove that there is a risk, but in the past it was kind of opposite. First, the chemicals or risk parameters had to be assessed before they have been released. Now they are released as a beneficial, but risk must be assessed. So if you want to comment.
I would just partly agree with you, because I think that the manufacturers of these formulation scientists, which sometimes also overlap with the guys who are publishing these papers, they are equipped with these. And I also read some papers about formulation science in the general, and they are using very sophisticated approaches similar to the development of new chemicals. So they have QSAR, they have a lot of instruments to characterize the surfaces of the carriers, the surface properties, the binding strengths, and all that stuff. So without that, they would not be able to develop these that approach. Just trial would be very expensive for them. They are not doing this. These guys from these companies, they very well know what they want to develop. And firstly, before starting any laboratory trials, they have designed the carriers in the computer. So I think they have a lot of knowledge. And it's just more about the communication between the assessors and these producers. So they know very correctly what they want to, what properties they are expecting from the carriers, from the formulations, and how to reach it.
But let me guess, they know it very well for the idealized environment, but not for the ambient environment.
And that
is what we are interested in.
That's the second part of my answer, that the bias between these scientists in the manufacturer side and scientists between these evaluator or assessors and the observers of the phenomenon, the bias is in the environment, because their goal is just to develop in the bottle the best formulation which has the desired properties, like increasing solubility and protecting before UV light and anything like that. And they do not care a lot about how to detect these particles, for example, in the complex matrices like sediment and water, which is something which these assessors and observers like we scientists want to have. And that's much more difficult. And in fact, there is just one technique or two techniques for the complex samples like soil or sediments, how to measure nanoparticles in these samples.
That's what I know.
Other questions?
I would like just to comment on that or add something. Wouldn't it be useful to put nanopesticides or nanomaterials under REACH?
The question is very simple, whether nanoparticles shouldn't be, or nanopesticides shouldn't be put under REACH.
The REACH is not distinguish the chemicals between according to purpose, I guess. It's so they are considered the same way as any other nanomaterials, which means that not too much. There are some specific assessment reports which evaluated the usefulness of REACH for the nanomaterials, and the conclusion so far I know was that in fact it's so broad, and the concept of REACH is so general that even the nano features of the chemicals are involved in that assessment, and that there is no need of any special guideline for the chemicals to be considered in REACH. But for the pesticides specifically, it's another legislation. It's a pesticide legislation in Europe, and there it's a bit different, because also the way and the concept of the assessment is a bit different than evaluating the chemicals in the REACH. And there are, for example, ecotoxicity studies mandatory for the whole formulation, for the product, including not only active ingredients but also the solvents and all other stuff. And in these testing, the nanoformulations, of course, will be also tested for the ecotoxicity, for example. But the question is if the results of such testing will be valid, because the methods are built for normal chemicals, not for the particles.
Any other questions?
A small question. Do you know something about changes of microbiota community in soil, depending on or related to carriers, especially polylactic or some sugars, polysugars?
No, I don't
well,
know.
the question is about the changes in microbiota in soils related to the carrier materials, like because they are polysaccharides and so on.
No, I don't know. There is no study about this. In fact, all studies which I mentioned of ecotoxicity were performed only in as aquatic studies. So in complex environment like soil, there was no ecotoxicity studies, and what you mentioned is in fact some ecotoxicity study with the microbes as an endpoint.
I think it's maybe more fertilizer or something, and again change community and maybe fertilizer.
Yes, and that's your question is also driving us to that. Also, the carriers themselves, even without the bound pesticides, can have some influence on living organisms, and this is something what hasn't been studied a lot yet. But it's also complicated from the methodological point of view, because these compounds are sometimes very similar to the natural compounds. So we were discussing with the colleagues from the group from Austria, from Wien, that probably it might be used some radio labeling to know the fate and ecotoxicity of the particles which we added to such complex systems. But it's methodologically not it's challenge, methodological challenge.
Salim?
Maybe just one last question also about REACH. We made some European regulations for all chemicals, which is quite strict, but definitely it's going really, really slow. But then meanwhile, we consider that pesticides, which are not chemical, they will not be considered in REACH, and we made some specific regulations for it. I don't know which one asks most requirements about the environmental fate, the toxicity, and so on. But what is the origin of these pesticides are not chemicals, but they have specific regulations?
Well, the question is basically about the difference, or the question is why in Europe we have regulations as a REACH, as a separate chemical regulations, and then pesticide regulation as a separate piece in our regulatory system.
If I get the question correctly, my answer would be that I think that the pesticides are also chemicals. They are also evaluated in REACH. So then just according to their use, that they will be used. And specifically in Europe, we are distinguished biocides and plant protection products. So if the chemicals are intended to be used as a plant protection products, there is some additional way how these chemicals must be authorized. So that the chemical is a pesticide and is going to be used as a pesticide does not mean that it was not evaluated by REACH and it does not bear all these CLP labels and all that stuff. So we have this data also for these chemicals.
And are all pesticides therefore included in REACH? I really didn't know.
Like, what do you mean?
The question is whether all the pesticides are in the REACH.
What do you mean by including the REACH? Like if they are authorized, or
they have been evaluated?
They must be authorized, of course. Firstly, according to REACH, and then they can be used as in plant protection products as active ingredients only if they are evaluated in the EFSA according to EFSA guidelines and listed on the list of approved active ingredients for the pesticides.
Are there any other questions? If not, also for the sake of time, thank you very much again for being here. Thanks a lot for fruitful discussion. Thanks to Jakub for a very nice presentation. And if you are interested, next week we will have another seminar at 9 o'clock, again on Tuesday, where our PhD student Lucka Štveráčková will be talking about her experiences from EFSA. She has a long-term stay there, so we can share another experience again on Food Safety Authority. Thank you very much.